I'm Vicki Risbrough. I'm a professor of psychiatry at UCSD, and I get the honor to introduce our next keynote speaker, Dr. Caroline Nievergelt. Caroline is also a professor of psychiatry at UCSD, and I've known Caroline for going on, I think, over 15 years. At this point, we sort of together, so to speak, in our training. So it's really a pleasure for me to get to have her come and speak at the Conte Center today. So Caroline is actually originally trained at the University of Zurich as a biological anthropologist. And I don't have a lot of time to regale you with any stories, so she can do that later. But please ask her about canoeing through Madagascar, catching lemurs and marmosets to collect DNA to establish a paternity. So she came to UCSD to work with David Woodruff, and then she moved over actually from biological anthropology to human genetics, working with John Kelso. And she was really on the forefront of looking at genetics behind treatment resistance in bipolar disorder. She then began to work more in the realm of post-traumatic stress disorder. That's where I met her in the marine resiliency study with Dr. Julian Baker and Dr. Dan O'Connor and Mark Geyer and that team. And she actually was the first person to publish a GWAS in post-traumatic stress disorder. She then started working nationally with multiple PTSD geneticists and developed the Genomics Consortia for PTSD. So this is the PGC for PTSD, Psychiatric Genomics Consortia. And this has really been a monumental effort. She is now past, I think, 10 years for this consortia. So this is a very mature consortia, and they've published a number of very high impact papers in Nature Neuroscience and Nature Communications, and they have a new one coming out in Nature Genetics that I think you're going to be seeing, which is really what are the genetic underpinnings of trauma and post-traumatic stress disorder. And just in terms of the impressiveness of Caroline, as I've seen her grow, she's really an amazing person. She has over 250 published papers. She is, I believe, the PI on, I think it's 5 now, R01s. I mean, she really has, really has shown, I think, the world kind of what psychiatric genetics can turn into with this kind of important effort. And I want to just state that this has now moved from national to international. So the consortium is now working, for example, with countries in Africa to collect data and information. So they're really going at this problem of post-traumatic stress disorder and the genetics behind it, at the at the international level. So I think you're going to hear a lot about that today. So without further ado, thank you, Caroline, for coming.
Thank you very much, Vicki, for aging us here publicly. And yeah, so I'm going to talk about Genomics Consortia for PTSD. I have 3 parts. First, I want to talk a little bit about genetic association studies and the path to success for the PGC. Then I'll talk about PGC for PTSD and then also mention our PGC PTSD working groups. Oops, sorry. So for a long time, we have known that psychiatric disorders are among the most heritable disorders. So what you see here is the heritability for 9 psychiatric disorders, plus 3 complex diseases, breast cancer, Crohn's disease, and type 2 diabetes, for which genetic dissection has been particularly successful. So what we also knew is that genome-wide association studies, or GWAS, are the primary driver of discovering common disease. And already in 2013, there were genome-wide association studies that were successful across a really broad range of disorders, diseases, and traits. So when we had the first large GWAS, actually in schizophrenia in 2009, we had 3,000 cases. We were very, very disappointed to find out that our first efforts were actually not successful. So what you're seeing here is a Manhattan plot where the x-axis along the you see the different chromosomes, and is that working? And each of these dots is a single nucleotide polymorphism, or SNPs. And you see the p-value here. And because there is millions of SNPs that we investigate across the different chromosomes, across the genome, we have a very strict correction for multiple comparison, which is at 5 × 10⁻⁸, and usually denoted by a red line. So, as I said, we were very disappointed, and so this led us to think that actually individual GWAS studies are just too small to identify robust and replicable associations, or any associations for that matter here. And so that was the beginning of the Psychiatric Genomics Consortium. That was really a surprising experiment, because it meant that people actually had to put their data together in part before they even had the chance to publish on their own data. So the consortium was led by a coordinating committee, Pat Sullivan, who is still leading it even now. So this started in 2009. We had 5 studies: ADHD, autism, bipolar disorder, MDD, and schizophrenia. And as Vicky said, I was working at the time with John Kelso on bipolar disorder, and I was very skeptical if this would work out. And we had the other working groups. The statistical analysis group is led by Mark Daly, which was very crucial at the beginning to have kind of this statistical thinking how we would go about this. We also have a copy number variant working group and a cross disorder working group that looks at across all of these disorders. So in 2009, we got our first round of funding, and then 5 years later, we expanded to 9 disorders for PGC2. We now had OCD, Tourette's, PTSD, eating disorder, and substance use disorder. And what we found, now the principle behind the PGC is the Recopily pipeline. And this was really what made the PGC successful, is that all the data was analyzed across all these studies and disorders with the single pipeline that had 4 modules for quality control, population stratification, imputation, and then analysis. All the data is stored in the Netherlands on the Snellius server, and every analyst that works with the data uses Recopily. So we get very standardized analysis, and that was a crucial part. So as I showed you in 2009, we had no genome-wide significant findings, and 3 years later, with 3 times the sample size, we had for schizophrenia 5 genome-wide significant findings, expanding to 25,000 cases, 62 hits, 36,000 cases, 108 hits, and so on. So it was really clear that increasing sample size accelerates discovery. So how does that look for the other psychiatric disorders? As you can see, the success extends to the other disorders. However, the trajectories are very different. So when you look at schizophrenia and the number of cases on the x-axis in thousands and the number of genome-wide significant loci, you see that schizophrenia in gray uses many less, requires many less cases to identify genome-wide significant loci compared to MDD, which is for us performs the worst, where you need about 400,000 cases to find the same amount of genome-wide SNPs. The other disorders are somewhere in between. In blue, you see PTSD. Also important is for each of the disorders to reach the inflection point, which is basically where you start to have a linear relationship between the number of cases and the number of loci you discover. And so this inflection point has now been reached for pretty much all of the psychiatric disorders that participate in the PGC. So you wonder what's next, how far do we want to go? Do we just want to keep expanding our sample sizes? So our goal is to reach a point where we are good enough, where we have discovered enough loci and genes that we can reveal the fundamental biology of a specific disorder, inform clinical practice, and deliver new therapeutic targets. So this is our goal. And so currently we are in PGC4. We have now also expanded, so our central idea is now to use a global cooperative network to advance genetic discovery. And we are not just focusing on US and Europe anymore, but we've expanded in Asia and are now also expanding in Africa. The coordinating committee still has more or less the same investigators. We also have now a very active outreach committee that disseminates our findings, a data access committee, so all the investigators can access the data and come up with their own analysis. We have expanded to, I think it's 12 disease working groups now. The newest groups are suicide and Alzheimer's. And we have expanded our working groups to also include cross-population and the newest PGC Africa group. So what did we actually find with all of this work? So I'm just showing you two examples from the cross-disorder group, which I think is very interesting. So our first large paper was in 2019, where we looked at across 8 psychiatric disorders with over 200,000 cases and 500,000 controls. And what we found is basically all the psychiatric disorders are highly correlated. There are 3 main groups. The mood and psychotic disorders with bipolar, schizophrenia, and depression, early onset neurodevelopmental disorders with autism spectrum disorder, ADHD, and Tourette's, and then disorders with compulsive behavior, which would be eating disorder, OCD, and Tourette's again. What we also found, interestingly, is there were 109 pleiotropic loci affecting more than one disorder. So among the strongest one was DCC, which is a gene involved in axon growth in early neurogenesis. We also found that the pleiotropic genes show heightened expression beginning in the 2nd prenatal trimester, and the pleiotropic genes play prominent roles in neurodevelopmental processes such as glutamate and calcium channel signaling. Our current work, which has not been published yet, looks at across 14 major psychiatric disorders. This is from a presentation at the World Congress in Montreal last fall from Andrew Götzinger, and he uses genomic structural equation modeling to model the extent to which disorders reflect latent factors representing dimensions of shared genomic risk. And what we found was a pervasive genetic overlap across all disorders, which is in line with the high rates of psychiatric comorbidity. We have 5 main latent factors. We have compulsive disorders, serious mental illness, neurodevelopmental disorders, internalizing disorders, PTSD, MDD, and anxiety, and substance use disorders. And these 5 factors reflect basically an etiologically useful way to understand the shared genetic signals in psychiatry. So these are our current findings, a paper that will probably come out in half a year or so. And one of the advantages and also shortcomings of the consortium work is, of course, In order to get this data together, all the working groups are willing to share their data with the cross-disorder group before they actually publish. So our PTSD data that's just about coming out now in Nature Genetics, we gave it to the cross-disorder group 2 years ago. So this data is based on data that has not been published yet, but the agreement is that the cross-disorder group has to wait until every single group then has published before they can publish. So the paper is written and we're just waiting for the last groups to publish their primary data. So I want to talk a little bit about the Psychiatric Genomics Consortium for PTSD now. So we were asked in 2013 by Thomas Lehner, who was leading the genomics branch of NIMH, to get together and brainstorm if we could come up with a Psychiatric Genomics Consortium for PTSD working group. And at the time it was hard to convince people that PTSD is actually a genetic disorder. So that wasn't not a thought that everybody believed in. So it took us almost 2 years to finally get funded, and we were very fortunate to have private funding from the Cohen Veterans Biosciences that got us together for face-to-face meetings, usually twice a year, and they paid for investigators across the globe to come to the meetings, and they also paid for genotyping of some smaller cohorts that couldn't be genotyped by the investigators otherwise. Currently we have 4 chairs: Kerstin Koenen, who is an epidemiologist, me as a statistical geneticist, Kerry Ressler, his focus is, as you know, in neurobiology, and then we originally we had Israel Lieberson, but now he is now followed by Maurice Stein at UCSD, who is an expert in PTSD phenomenology and also a co-investigator on MVP. So challenges compared to other PGC disorder groups are PTSD is a very challenging disorder in terms of the diagnostic criteria, as you all know. And basically, there is over 600,000 ways to have PTSD, which leads to a high heterogeneity in the symptom profile. So in addition, the PTSD prevalence is directly related to the severity of the traumatic event, and certain events such as rape or direct combat have a very high risk compared to other events such as accidents. So for us, the task was to harmonize the phenotypes. We realized we had phenotypes from different studies across 3 DSMs. The phenotype was either lifetime or current PTSD. We had different instruments such as clinician-administered the CAPS, self-reports such as the PCL, over 10 other instruments. So it was quite a challenge. So in order to move forward, we said, okay, to each investigator of each study, could you please just tell us who are your cases and who are your controls? And so we moved with that. And in addition, compared to other PGC working groups who are predominantly at the time of European ancestry, as you see on the left, we had different studies with a high diverse genetic ancestry. And so why is that important? So this is my teaching slide, so I would like to walk you through this because it's really important concept that influences all the analysis that we are doing. So what I'm talking about is the linkage disequilibrium. And linkage disequilibrium, or LD, is the non-independence of alleles at nearby markers in a population because of lack of recombination between the markers. So what you see is different stretches of the chromosomes here, and over time, because of recombination, as you probably all remember, these stretches become shorter and shorter, so these are the haplotype blocks. And every mutation on a haplotype block is inherited together with the other mutations. So this is basically a unit. And so Of course, over time, the stretches become shorter, and because of the older origin of the African populations, the LD across the human genome is generally lower in West Africans than in Europeans and East Asians. And so this is important because when we perform a genome-wide association studies, so if you look at these are the stretches again of a chromosome with the LD blocks here, you can see compared to the Europeans, the Africans have much shorter blocks. And then these circles, the field circles, are actually the SNPs, the basically mutations along the chromosome. And so what we are doing in the GWAS is we are trying to have markers that are only taking one marker, taking one haplotype, one haploblock. So we don't need so many markers to genotype. We basically reduce to these markers. And so what you're finding is that when you actually have a functional mutation here in red and blue, it is perfectly tagged by this marker that's genotyped in the Europeans, but because there is a different LD structure, in the African population, this marker would not be associated with the disease because it does not tag the area where the gene actually is. And so if we combine African and European samples, we dilute our result because now we have half of the population would actually have the association and the other one would not. And so that is the reason why it is so crucial to think about ancestry very carefully. So that's what we have done. All the analysis in PTSD has basically been performed by our stage 1 and stage 2 analyst Adam Mayhofer in my lab. And so what we did is, because a lot of our data is actually not shared at the genome-wide or individual level, we have everything we do has to be portable. So we have to make pipelines that we can share with investigators who have data that cannot be shared with us and only share the summary results. So we made a portable pipeline that was able to use ancestry informative markers and importantly ancestry reference populations and come up with a way that we could do a consistent ancestry calling across the different studies. And so then what we did is we made a very homogeneous European group that basically conforms to the other working groups in the PGC. And we analyzed that is usually by far the largest group. And then we looked at two-way admixed individuals that have African ancestry or Native American ancestry, so our Hispanic populations. But we excluded people who had more than two-way admixture or were from other ancestries. And so our first findings were with 20,000 subjects, we had no genome-wide significant findings in phase 1. 2 years later in phase 2, we had 10 times as many subjects. We had our first genome-wide significant findings. However, when you look at these Manhattan plots, you can see that our findings are very limited. We didn't have much power. And so it was clear that we needed to still expand our sample size. So what we did find and what is still holding up from this first paper is we found significant genetic correlations between PTSD and the other traits and disorders, such as psychiatric traits, anthropomorphic traits, reproductive traits. We have correlation with negative correlations with years of schooling, age of first birth, subjective well-being, and then positive correlations with MDD, depressive symptoms, waist-hip ratios, and interestingly, asthma. So the goal for our next round of analysis was, as I said, increasing the sample size and improve the phenotype characterization. But we also wanted to expand to CNV analysis, optimize the contribution of diverse ancestries, and do sophisticated post-GWAS approaches to identify functional consequences and use polygenic risk scores to advance causal inferences. And so, as you can imagine, we, in order to yet again increase sample size massively, that takes a very long time. And so, while we were working on this, we wanted to do something else. And Adam Mayhofer, who at the time enrolled in a PhD program, he came up with some ideas for his PhD. So as I said, we were very underpowered for locus discovery with our FREEZE-2 data, but we still had data on over 30,000 PTSD cases with significant heritability. We were able to do genetic risk prediction in outside samples, and so we knew that the data had a lot of information, just not yet for locus discovery. So we remembered that we not only had our case control scenario, but that we actually had collected data of over close to 200,000 subjects with quantitative PTSD symptoms, as well as self-reported trauma exposure. So Adam came up with a way to enhance discovery for PTSD through integration of quantitative phenotypes and trauma exposure information. And so the idea was that it was shown by Naomi Ray that quantitative outcome measures can actually improve power in GWAS. And that is not only in case control scenarios, but especially in population studies, and which was what we predominantly had. At the same time, there was a program coming out to perform multi-trait analysis of genome-wide association, where you could use summary statistics from genome-wide association studies of different traits with, importantly, they could also have overlapping samples. And so the key idea was that when GWAS estimates from different traits are correlated, the effect estimates for each trait can be improved by appropriately incorporating information containing in GWAS estimates of the other traits. So Adam was very excited because he did not want to go back and work with all these different trauma exposure measures in all these 90 studies. So he was like, oh, that's great. So he did one GWAS in the UK Biobank on 130,000 subjects on lifetime trauma exposure, which was significant, showed a significant heritability of 7%, and the genetic correlation with PTSD was just perfect at 0.6 to be usable to use for MTAG. So he integrated this quantitative symptom score GWAS with MTAG of trauma exposure, and what he found is 8 genome-wide significant loci. So that was basically the first time it looked like we had some decent signals, and this is in exactly the same data as I showed to the right from FREEZE-2 Europeans. So this is the same subject, the same genotypes, just analyzed in an enhanced manner. In addition, he also looked at copy number variants. He worked with Jonathan Sebat, and what CNVs are is basically stretches of the chromosome which are duplicated or show deletions, and you can use the intensity of the SNP signals on the arrays to detect these stretches by seeing that the intensity, if you have a deletion, is lower than from the other markers, or higher if there is a duplication. And so he looked at the overall genome at CNVs, compared CNVs between cases and controls, and did not find any differences. But when he focused on CNVs known from neurodevelopmental disorders, he actually found significant CNV deletions were overrepresented in cases compared to controls. And when he looked in detail, there was a known duplication on 22q, as well as a very known deletion on 15q that were significant for PTSD. So that was the first CNV paper in PTSD. And finally, we wanted to work on a little bit more on admixture, mostly because that is my background as a population geneticist. And so I was excited to look more into this. And what you see here again is the same picture where you see typical African American individuals who are on average 20% admixed with Europeans. However, there is a very wide range between the subjects. And typically we just look at the average admixture for an individual and adjust for that with our PC analysis. So, of course, when you just look at one individual, you see that across the different chromosomes, and you have the 2 copies of the chromosomes, you see that there are stretches where the origin is from both parents of African origin, but then you also see stretches where you have 1 copy of European and 1 of African ancestry, or you can also have 2 copies of European ancestry. So just taking the average is probably not the strongest approach. And so we worked together with Elizabeth Atkinson and Ben Neal's lab to come up with the method to actually look at local ancestry in admixed individuals and incorporate that into our analysis. So the pipeline, again, a portable pipeline to call local ancestry was called Tractor, and here is an example how we were able, in the example of total cholesterol, for example, we looked at a regular standard GWAS analysis, and at the top hit would be here, this SNP close to DOC6, but then when we used our Tractor method, we could fine map better, and now we found that the signal actually moved and is now here, and this gene ANGPTL8 is a much better candidate for total cholesterol. So that's an example of this method, how it can help in fine mapping. So finally, we have now reached, we have now assembled enough studies, over 90 studies, over 1 million subjects with over 140,000 PTSD cases. We called that phase 3. The study is now in press at Nature Genetics, and I want to show you some results of what we have now. In terms of phenotypes, how we were able to get to these sample sizes. In addition to the clinical studies, we now also worked with large EHR data sources where we have less information about symptoms on PTSD. We're just using ICD codes, but we have extensive information on comorbidities. We had an administrative supplement, and Carmel Joy, who works in PsychEmerge, is leading the EHR working group and helped us for a consistent defining of PTSD. We have a broad and a narrow definition, but for the GWAS we worked with the broad definition. We also have another data source now, led by Maurice Dean and Joel Gelernter. We have the MVP, the Million Veteran Program, which is an awesome data source. In their first paper, they used over 200,000 subjects, and 40,000 had PCL4 with PTSD symptoms. So that's a very unique and homogeneous data source. So since these studies are also different, first we wanted to see, can we actually combine these different data? So we used a method called MIX, or a univariate Gaussian mixture model, where we can look at the genetic architecture of different studies or GWASs. There are measures such as polygenicity, which describes the proportion of causally associated SNPs, the discoverability, SNP-based heritability, and can also look in bivariate models at the polygenic overlap of complex traits, which is irrespective of their genetic correlations. So this is a very informative modeling, and basically what we found, comparing our quantitative symptom score, freeze 2.5, to the homogeneous MVP data source and our 10 EHR sources. Oops, so here on the right, we found that very similar genetic architecture across the data sources. We had high genetic correlation between all subpairs, and we have no evidence for data source-specific variation. So we felt comfortable that we could combine the data, and in our phase 3 analysis plan, the strategy is still the same. We first stratify by ancestry. Each study gets stratified by ancestry, and then we do a meta-analysis across the studies within ancestry, and then finally a meta-analysis across the different ancestries. And so, first look at this new freeze. We were really impressed. We now have in the multi-ancestry analysis 85 genome-wide significant loci. Overall, in looking at all the other ancestries, we have 95 unique loci. Here you see the Manhattan plot of the multi-ancestry combined with the European ancestry, and you see that the pretty much the same signals between the two analysis. So in terms of the overall genetic architecture, we have now a SNP-based heritability of 5.3%. We do have no significant differences in heritability between the sexes anymore. Looking at the other ancestries, we don't have signal yet, so we still need to expand. And then, of course, we had extensive post-GWAS analysis, including a large team of statisticians and people with expertise in biology. We do a careful genetic characterization, looking at the architecture, genetic correlations, gene set analysis, gene tissue analysis, as well as extensive gene prioritization using gene mapping, functional annotations, fine mapping, TVAS, EQTL, PQTL, and also drug targeting and polygenic risk score analysis. So I'm just showing you very few examples. Here, this is probably what people are most interested in, is the actual 175 significant genes, including DCC, that was the, oops, you can't really see that on this, the DCC, that was the pleiotropic gene found by the COS disorder. We also have FOXP1, FOXP2, a lot of genes that are known, including DRD2, former candidate genes, that people still remember from the olden days. In a gene set analysis, we find 12 significant gene sets, including biological processes, functional, molecular function, and cellular components. All these processes make sense. Looking at the transcriptome-wide association studies and summary database Mendelian randomization in brain, as well as blood and proteins, we find different signals across the different analyses, as well as consistent signals. So the big challenge was, how can we actually combine this data, make sense out of it? So we came up with a gene prioritization way, where we basically looked at the 81 genome-wide significant loci, and then we prioritized the mapped genes based on weight of evidence across the functional categories. And then we based these genes into tier 1 and tier 2 genes. And so that way, we prioritized the genes. And what we found is that it actually worked. So when you look at this SYNGO, this is a sunburst plot where you see PTSD-related genes tested for overrepresentation among genes related to synaptic terms. You see no significant findings of all the genes that we had before prioritization, but after, we actually see enrichment in the synapses. So this is kind of a proof that our method actually worked. Okay, so the last example of this freeze too, I would like to show you the polygenic risk scores that we now have. So looking at this plot, you see the polygenic risk score explains 2.8% of the phenotypic variation in PTSD. So that's not much, you would think, but it's highly significant and much, much better than we did earlier when you compare the yellow dots from freeze 2 to the blue dots from freeze 3. And so the highest quintile now here, you can have a 2.8-fold higher odds of PTSD compared to somebody with the risk profile in the lowest quintile. So this is very successful. However, when you look at subjects of African ancestry and you use the European ancestry results, you see that it performs much, much poorer. So this is the difference. And a little bit better for individuals of Native American ancestry, so the Latin, Hispanic populations. So what we have to do is clearly we have to go and expand data collection in Africa to actually make this equitable approach. So I just in my last minutes would like to talk a little bit about the PTSD working groups, of which I am particularly proud of. So at the core of this figure, you see our core genotype data and our core phenotype data. And when we started working with all these investigators, it became apparent that there is so much expertise and interest among us. Some people were interested in imaging, in different phenotypes, physical health, TBI, substance use disorder, as well as we also had interest in complementary omics data, such as copy number variants, epigenetics, and systems biology. And all of these working groups have been leveraging the genotype and phenotype data, and the working groups that are in shaded boxes actually are funded or have been funded for quite a while now. So I show you 3 examples. The first group that actually got independent funding is the epigenetics working group. We started in 2016, and we are working on our second freeze. We have over 5,000 subjects that have genotypes as well as epigenetics data, costing 23 different studies. We have military trauma, civilian trauma, and not just lifetime PTSD, but here we're looking at current PTSD. And then we have the Enigma PGC PTSD working group, started a year later, led by Raj Murray and Mark Logue. We are focusing right now on the genomic architecture of functional brain networks in PTSD. We are one of 55 working groups, so Enigma is a great resource in terms of methods for us as well, and very interesting to follow. And then most exciting for me and kind of coming full circle is we now just recently were able to get funding for African ancestry working group. We have a grant and actually enroll subjects into studies in Uganda and in Kenya. And yeah, hopefully this will then contribute to better polygenic risk scores and different findings. With that, I would like to thank the funding agencies and IMH and especially Cohen Veteran Biosciences and the many, many, many investigators that work on these projects. Thank you. Yes.
Thank you so much. This was so interesting to listen to. I'm curious, now that you're starting to identify some of these genome-wide significant loci and things like that, what do you see as the next steps as far as how that might move the needle in the way we understand and treat PTSD?
Yeah, that's a very interesting question. Of course, one of the things that I've pointed out is that there is very similar signatures across the psychiatric disorders. So I'm not sure if this changes much how we treat PTSD as so much as how we would look at the whole spectrum. Yes.
The significant loci that you've identified for PTSD, do you know how many of those proteins, related proteins, not genes, but the transcribed into proteins, have cofactors such as metals or vitamins? What percentage?
No, I do not know that. We just identified these loci, and we haven't been able to characterize all of them yet. Yes, what a good question.
What?
So when you answer the question, can you just repeat that question?
Yes, okay. Yeah.
Oh, right, okay. Any other questions?
Tally?
Oh, Tally?
So, in the previous talk and also some of what you said, it looked like many of the factors for her early in life, essentially transcription factors that are hormone blocks or something that is involved more in the brain fundamentally. Now, in PTSD, we sometimes think about it, disorder that happens after adversity early in life or late in life. Is the group able to find a different genomic architecture or signature in PTSD that happens after early life versus adult life? Would you agree?
So the question is if we would find, if our group would be able to identify different signatures if a trauma occurs early in life or later in life. And I guess with the work we are doing, that would be it would be difficult, right? Because we have lifetime PTSD, so we don't even really know when the trauma happened. But the epigenetics group, who has the current PTSD assessment and more detailed information about childhood trauma and adult trauma, that would be where we would do this kind of work. Yeah. Yes.
Hi, great talk. I have a question about the clustering of the genetic markers by diagnostic groups that you showed. So I'm wondering if it's more useful to sort of drop the diagnoses and start thinking about symptoms and say, because two people with major depression may have different symptoms, but genetically sort of we're looking at them related to the diagnosis of having depression. Same with PTSD, same with ADHD. And so I'm just wondering what you think about the idea of looking at people that have common sets of symptoms, forget diagnosis, and then looking at the genetic markers and clustering in that way.
Yes, so whether that was a long question. Vicky, what was the question?
So the question was basically, instead of using diagnoses, would you use like symptom clusters or sort of item or factors, right? So sort of using, say, a huge PCA kind of or factor analysis where you'd have better factors that maybe give you some information that's not the diagnosis, but is some sort of core symptom cluster that then is better correlated to the genetics. Is that?
Yeah, or the genetics telling us something about the symptoms. In this case, maybe it's telling us something about a disorder identifier, but...
Right. I mean, if you imagine how this started, right, the cases and controls, especially for the other disorder groups, are just case control definitions. So then you do the GWASs on that, so we can't really go back. We don't have the information, right? So the RDoc would have been a better way to go about that, because then from the start you would actually collect this information. So it's not really possible. Whether I think it's a better way, I mean, I don't know. One would have to see, right?
Didn't Murray do that? Like Murray Steen, he did with MVP. He did the clusters.
Yes, for PTSD, the MVP group did that. They looked at the B, C, and D symptom clusters and did not find they were genetically so highly correlated That was actually our idea to do that for the whole consortium, but based on their findings, we abandoned that idea.
So
Yeah.
far, the clusters haven't really given you any unique
No,
markers.
no.
Yeah, that's a pretty good. So far, the clusters haven't given any unique markers, so they're back to disorder specific.
Which is interesting if you're thinking about treatments based on genetics.
Yeah.
Okay, thanks.
Okay, maybe one more question and then we can take a break. We're all done? Okay.
Done?
All right.
Okay,
Thank you,
right, thank you.
Caroline. Okay, so we have time, like a 5-minute break, I think, right? And then we have, yes? And then we have another speaker at noon. 7-minute break.
Okay, folks, we are going to come back, return to our regularly scheduled programming, so please come on back, have a seat, and relax. There's only one talk between you and lunch. Okay. All right, our next speaker is our second of our UCI speakers. This is Dr. Stephen Schuler, who's an associate professor in the Department of Psychological Science, and I guess I'm just finding out now, Department of Informatics as well. He'll speak to us about digital exhaust and digital interventions, harnessing big data in mental health technologies. So with that, Dr. Schuler.
Awesome. So I'm going to be talking about big data in a very different way and thinking about how some of the different interactions that we do with devices that smartphones and other types of things, as well as different digital interventions, a feature that we called normalized entropy. So this was the regularity in terms of which they're moving through geographic space, normalized on a circadian cycle. So looking at, does a person leave their home at the same time every day? Do they make it to work at the same time every day? Do they have a regular pattern in terms of how they're going to different places? And what we found is that people who had a greater level of regularity tended to be less depressed, that this habits and these patterns sort of formed, and that was actually the strongest predictor. And so this was a very early study that we published back in 2015 that looked at this. We've done various different studies using the same sensor acquisition platform and looked at the same features and have mostly found that these location-based features and movement through geographic space is the most predictive. I will say we've also done some work in some different types of populations, including youth experiencing homelessness in Chicago. And in that sample, we did find that none of the same features or feature sets that we used were actually predictive of mental health states. Instead, the two things that we found were most predictive in that sample of youth experiencing homelessness were battery life and access or connection to Wi-Fi devices. And at first I was like, battery life, what's this about? But if you think about it, for a lot of us, battery life is something that is, there's no noise in this data. We plug in our phones at night, it gets charged, and at the end of the day, it's lower. But for individuals experiencing homelessness, this was actually a pretty big marker of when their life was more disrupted, when their life was more challenging, that they weren't able to always consistently charge their phones. When I was doing focus groups or other work with this group, usually the youth would come in and they'd look around for the different sockets to start charging their phone, because it's like they knew they'd be in the same place for an hour and a half and it'd be safe to put their phone somewhere and someone wouldn't walk off in it. So I do think, although this shows some promise, I think one of the big challenges that we have to think about is how such algorithms or how such models can scale and be relevant to diverse populations, especially populations that may not match a lot of the same characteristics of individuals who traditionally make their way into research settings. Okay, so now I'm going to shift gears and talk a little bit more about how we can think about using technology, not just to understand people, but actually try to help them, move a little bit more in the intervention realm. And I will say, in the context of digital mental health interventions, there is a lot of research that's accumulated over the past 2 decades that show that digital mental health interventions are effective, especially for common mental health disorders like depression and anxiety. Now, what we find, though, is that the most effective digital interventions are those that blend some type of technology component and a human component. So digital mental health, when provided by itself as a standalone intervention, usually shows small to moderate effect sizes, but interventions, when they're combined with human support, actually have moderate to large effect sizes that are actually on par with the frontline treatments that we usually deliver in terms of psychotherapy. One thing I think that's really interesting as we think about opportunities for personalized care and the opportunities to really use big data from these types of digital interventions to drive more effective, more personalized interventions, is this human support might be most important and most impactful for those most in need. So this data here on this table at the bottom comes from a large individual patient-level meta-analysis that Irina Karatoki led. And what you can see here in this table is the effect sizes and effect size comparisons between guided and unguided programs, as well as in comparison to treatment as usual, at different levels of, initial levels of depressive symptoms. So you see at the bottom here, see if I can use this right. If a PHQ-9 around an 8, we see that there's very little difference between the guided and unguided programs. In these cases, you don't need human support to get the same benefit out of these types of interventions. But as the symptom level increases, PHQ-9 of 10, 14, 25, you see more and more differentiation between the guided and unguided options, such that individuals with the highest level of depressive symptoms actually are the ones who are most likely to need this sort of human support. And so I think one thing that we are trying to be really thoughtful about as we develop and try to scale some of these different digital interventions is how we can most efficiently and effectively use that human support to help support those in need. Okay, so I'm going to talk about one intervention program that we developed, and this is a platform called IntelliCare. And many of these digital mental health interventions will make a platform that will have a variety of different aspects. We decided that building one platform was too easy, and so what we just did was we built 13 different mini applications, and each of these mini applications was based on a specific sort of treatment strategy, mostly coming from cognitive behavioral therapy, but adapting some other ideas as well. And they were really meant to be very simple treatment apps. You could use them quickly, you can use them efficiently, but really focused and targeted on a specific technique. So for example, we had a cognitive restructuring app that taught people the skills around cognitive restructuring. We had an app that was focused on behavioral activation. We had a goal-setting app. Just as a little indication or a demonstration of what one of these apps might look like, this was one app that I developed. It was one of my favorites. It was called My Mantra, and I thought about this as like the Pinterest of cognitive behavioral therapy. So what you would do is you would come up with a mantra, a self-affirmation phrase. It could be something like, I'm strong, I'm resilient, I'm cared for. And you'd be able to look through your phone or through the internet to find different images or different pictures that captured or represented that mantra. And then the My Mantra app would push to you throughout the week that image with that mantra at different times. It would give you reminders for it. So this is just an example on the right of some of the mantra boards that could be created. I'm grateful, I'm strong, and then it would push those to you at different times. And so effectively what you would do on the app is you would create a self-affirmation phrase. You would go through your pictures or other pictures you could find on the internet. We had some template ones in here as well. And you would create those mantra boards that would get pushed to you. As mentioned, we had a variety of different apps that were present in this IntelliCare suite to be able to sort of think about how we might be able to tailor sequences or collections of these apps for individual people. So the first stage that we did in terms of developing this IntelliCare suite and being able to collect some of this information that we would need to later tailor these interventions is we actually just did a public deployment. We put these up on the Android App Store, and we allowed individuals to download and install them. And in the first 2-year period, we got 80,000 installs among 76,000 unique users. I'll just say that the average use of these were aligned or maybe a little bit better with the common usage we see across mental health apps more broadly. Now, that's not to say all of them were great. And actually, Me Locate was an app that we had that was using geolocation features to be able to push sort of targeted interventions, and we did not see that one used that well. So we actually then retired this app before we moved to later field trials and subsequent randomized control trials. But this allowed us to collect a lot of information about how people use these, what were the engagement patterns, what was the relationship between some of these different apps. We then moved on to do a couple different studies of IntelliCare, again, to build up some of the evidence base for both the intervention program, but also to collect some of the different data we would need to be able to lead to these tailored, personalized interventions, which I'll talk more about. So our first study was a single-armed 8-week coached field trial of IntelliCare with about just under 100 participants with elevated levels of depression and anxiety. You can see that over time, we did see reductions of depression and anxiety that were on par to traditional treatments. So they started at a PHQ-9 of around 12.5, and after 8 weeks of receiving IntelliCare, we had PHQs that were below symptom threshold of about 7.5, and we saw similar reductions in anxiety scores. So I mentioned there was a coached intervention, and to go back to the point I made earlier about human-supported interventions being the most impactful and having the largest effect sizes. We used a model of human support that was based on a model that I developed called the efficiency model of support. Effectively, what the efficiency model did is it had the coaches look for different aspects in terms of how an individual might fail within a digital intervention and try to intervene on these different points. And so we identified 5 different failure points. It could be a usability problem, just technical problems in terms of using the actual platform. They don't know how to use it. They don't have sufficient tech literacy. It could be an engagement problem. They're just not using the app, so really encouraging people to engage. It could be a fit problem, that the specific intervention techniques are not relevant or they don't see the relevance for their life, and so we have to sort of make those interventions more relevant. It could be a knowledge problem, especially because these are based on cognitive behavioral therapy principles that we're often trying to teach skills or knowledge through these different interventions. Or it could be an implementation challenge in terms of that they know the skill, they know how to use the skill, they're just not applying it to their lives. So they're using the app, but they actually have to take these skills and use them out in the world. And so the coaches were trained to sort of interact or intervene on these different sort of failure points. And one of the ways that we were able to support the coaches in doing so is we had a coached dashboard that you see an example of here on the slide that actually provided the information to the coaches around how people were using it, what were some of the challenges they were facing, what were the apps that they were loaded on their phone and recommended to use in that specific week. So I mentioned earlier that one of the reasons we did this public deployment from the start was we wanted to collect data about the engagement with the different apps, how they were using them, which apps they were using. And we wanted to do that because when we were moving to our final randomized study of IntelliCare, one of the things we wanted to do was have a recommendation engine that would take those different now 12 mini applications that were present there and be able to make recommendations for an individual person about which app might be the best app for you to use. And so we had a hub app that would actually go in and give you recommendations from this small sort of app store of different IntelliCare apps. So it would say, for example, this week, you should download Aspire, Daily Feats, and Day to Day and try to use those applications to see if they work for you. So it would take the different sets of apps and provide sort of a personalized recommendation. Okay, so we moved on then and did a randomized control study of IntelliCare, and we used a 2x2 factorial design in which we compared coaching versus self-guided. So some individuals got IntelliCare along with a coach. Some people just got the apps to use by themselves. As mentioned, we predict that the coached version should be more effective. But we also compared algorithmic recommendations versus no recommendations, essentially allowing people to have these personalized sequences of IntelliCare apps that they would get recommendations on a weekly basis about which app they should be using that week. We used the coaching based on the supportive accountability and efficiency model of support that I showed earlier. And this was an 8-week intervention, and we looked at primary outcomes of reductions in depression and anxiety as well as engagement. So what did we find? In terms of the clinical outcomes of IntelliCare, we found that if we looked at the coached versus the self-guided versions, there was a slight benefit for the coaching. This was significant for anxiety and not quite significant for depression. We also found that for depression, the recommendations were more effective, had larger decreases in depressive symptoms than individuals who did not receive recommendations. So essentially, the coached recommended version was the best performing condition, but actually receiving the recommendations was able to overcome some of the benefits that we saw from those coaching. So perhaps one thing that was going on here is that the coaches are doing some of that personalization, that recommendation through the support that they're providing, and that support might be able to be provided through these recommendations that come from these machine learning algorithms and recommendations. Okay. So I also want to talk a little bit more about other potentials of the big data that can come from this type of thing. And so what we also noticed is that people tended to build their own library of IntelliCare apps over time. We did find that most people were following our recommendations of trying to use a couple of these apps each week, and that over the time in their study, we saw that they were cumulatively downloading more and more of the IntelliCare apps so that they would kind of use 2 at the start and like slowly build up to all the different apps in the different in the suite. We also found that there were different types of users of IntelliCare. They tended to go into different clusters of usage, and we found sort of 4 clusters that were indicative of the use that we saw in the use of the IntelliCare system. We found people who are low users and high users. This is not surprising. We see these patterns in a lot of different digital interventions, that some people tend to be the high engagers, the super users, other people use things less frequently. But also speaking to the benefit or the aspect of our sort of personalized treatment model, we found that there were kind of 2 clusters of app use, of the different apps that people used and then benefited from. And we referred to these people as sort of daily feats users or day-to-day users. And the reason was because those were 2 of the apps that were indicative of the most use, but also daily feats and day-to-day represented 2 very different ways of using applications. So daily feats was our goal-setting app. Essentially what you did in daily feats is you took a PHQ-9 and GAD-7 at the beginning. It looked at your level of severity of depressive symptoms and anxiety symptoms, and it gave you a list of different things you could accomplish that day. If your symptoms were quite high, those accomplishments were very simple things. Get out of bed, make good choices. And in fact, this was a really popular app. We heard from a lot of our users that they really enjoyed that we framed things that they really struggled with, getting out of bed, getting dressed, taking care of themselves, as feats, because that's what it felt like for them. When they were able to do that, they felt like they accomplished something, and to have an app that they could check that off their to-do list and get that benefit and that encouragement from the app was really useful for them. So we had these daily feats users. These apps were sort of more indicative of sort of short-term regular use. And we had our day-to-day users. Day-to-day was an app that had a couple different treatment strategies in it. It had behavioral activation, cognitive restructuring, gratitude from positive psychology, as well as problem solving. You would have a short lesson at the beginning of the week, and they would push you content each week. Day to day, it would give you a short little thing that would reinforce how to be grateful that week or how to engage in problem solving. So this was a little bit more pushing information to people rather than having them do something actively each day. So these were apps that tended to not need as much active engagement from the individual, but was pushing stuff at them, like day to day. So these were the 2 types of users we saw. And we saw that if we looked at the relative app use across these clusters, we did see that individuals in each cluster tended to have higher use and higher benefit from the different apps that were present in the related clusters. We also found that recommendations increased the use. So when individuals did receive this recommendation from the algorithm in that week, it did increase the use of that subsequent app in the following week. So we found that the algorithm was sort of producing the engagement benefit that we would hope to see. Another thing that was really interesting when we think about this from a big data perspective is we also, because we had the coached interactions between the individual and their coach, we looked at the different language that was shared in those messages. So what was the coach saying and what was the individual saying? And we did text analysis to be able to look at how some of the different language within those communications related to different aspects we saw in terms of the engagement across those high and low usage clusters. So we found that the low usage cluster was less engaged, but did ask for suggestions and follow recommendations. So they weren't as actively engaged with the coach, but they did ask for different things that were present and follow recommendations when they were given. And the high usage cluster communicated problems both with the apps and their life, as well as different positives that they received from the different applications that were present. So I think this is another sort of aspect in which we saw that we could use some of the information that came from the language or the content to better understand how a person was using these tools, as well as maybe speaking potentially to the way we might be able to intervene and support engagement. Okay, so just a couple thoughts, and then I'll kind of move into some conclusions here. But the future of big data and digital interventions really require these two areas I presented to come together. We need to think about how we can better use some of this information about understanding people for assessments and digital phenotyping, and pairing it with opportunities through digital interventions to create better personalized and more impactful interventions. As technology becomes better at being technology, as we see advances in things like generative AI and large language models, we have to become better at being humans. We have to think about what are the aspects that humans bring that are unique to the equation, and what can technology do to really sort of expand our capacities and our capabilities. How can technology enhance care rather than replace it? And how do we allow people, especially people with necessary expertise, to do more of what they're good at? Technology also gives us an opportunity to better understand what happens in therapeutic processes. As mentioned, a lot of the challenges with understanding psychosocial interventions, they are complex, multi-factored interventions. And I think despite us having years of research in terms of psychotherapy, we still don't really understand what's going on in that psychotherapeutic process. And lastly, I really think we have to make sure that data does not speak for itself. We really need to consider how to use this data to create intelligence. And we need to make sure that we allow individuals like ourselves, clinical experts, experts in mental health, experts in really understanding the phenomenology of these disorders, to help inform some of the features that we're creating so that they are clinically meaningful and clinically useful. In service of kind of speaking to this point about opening up the black box of therapy, this comes from a paper that was published in JAMA Psychiatry back in 2019. And what they did is they took a internet cognitive behavioral therapy platform, a digital treatment that sort of had the principles of cognitive behavioral therapy. And because it's all digital and because it's all tracked and because there's digital exhaust, they were able to look at, well, what were the things that went on in that therapeutic relationship that actually related to better outcomes? What was it that drove change? And they used machine learning models to look at that. And you can see that some things up here, like therapeutic praise, planning for the future, perceptions of change, these were things that favored improvement. Things that were not related were kind of the in-between stuff, like the bridges between different content, saying hello, saying goodbye. Setting homework, not so useful. Reviewing homework, much more useful. So all you burgeoning therapists and clinicians, homework is only useful if you actually follow up on it. You can't just give people homework, they actually have to do it. You have to integrate it into the therapeutic process. And so we can use some of this digital exhaust and this machine learning algorithms to better understand what's going on in therapy, and I hope build better interventions. This is an example of a study we're doing from a recently funded R01, and we are doing an evaluation of digital cognitive behavioral therapy for monolingual Spanish speakers recruited from safety net primary care settings. And in doing so, we're looking at the impact of individuals receiving a unsupported digital cognitive behavioral therapy platform, just standalone digital cognitive behavioral therapy, which, like I said, is effective, but we usually have small to moderate benefits. And we're comparing that to individuals who are receiving peer-supported digital cognitive behavioral therapy. We're training individuals from the local community who are culturally share a cultural background with the individuals in our study or the patient population. These are not trained professionals. They're not community health workers. They're just people from the community who have identified mental health as something that is needed in their community and want to sort of figure out something that they can do to help support it. So we're giving them light training to be able to better support and engage this program. We're also looking at some implementation issues and doing this in the context of a hybrid type 2 effectiveness implementation design. This is not our work, but this is work from another team that's using the same platform. So in our study, we're using the digital cognitive behavioral therapy platform of SilverCloud. And SilverCloud has teamed with researchers from Microsoft, and they've actually looked at how they can make better, smarter technology-enabled services by using artificial intelligence to help provide more support to those supporters who are engaged in the program. So thinking about our community members who are trying to support this program, how can we allow them to use the information that's present there in sort of more actionable ways? And so what Microsoft did is they took data based on the improvement that people would be expected from this program, based on engagement and symptom improvement profiles, and they created sort of a smart dashboard for those clinicians that would tell them the likelihood that these people would improve based on their current usage and based on their current PHQ-9 and GAD-7 scores. So essentially you can see, you know, based on a PHQ score here, this person is likely to achieve reliable improvement. Based on their GAD-7 score, they're unlikely to achieve reliable improvement. So you might be able to think about ways you can encourage that person, that supporter, to help support the patient in doing things that might better address their anxiety. And this is something that we've been looking at in other sort of instances in how we can actually use AI to better predict and then tailor programs for specific symptom profiles and specific symptom needs. Okay, so just in conclusion, some thoughts and takeaways. Psychology is a human field, but that doesn't mean technology can't help us be better humans. I think something I get asked a lot is, you know, when are the robots going to replace us clinicians? No time soon. I think that's a really important thing to keep in mind. Technology is not about replacing clinicians. It's about scaling and improving care so that we can be better, faster, stronger at supporting those in need, supporting those who suffer from mental health disorders. Our interactions with digital devices and software speaks to our behaviors, GPS location, but also a lot of other research teams are looking at things such as phone interactions, typing speed and typing the way you type, as well as content, things that you say. And we can create better, tailored, personalized interventions from combining several ideas from the past. Modular care, measurement-based care, stepped care, tiered care. These are all ideas that have been around in psychological service delivery for a long time, but it's hard to accomplish in terms of the typical type of in-person human interventions that we provide. Digital interventions really provide this opportunity to better modularize care, better track care, and better understand what interventions might benefit specific individuals. And thanks so much for your time. I really want to acknowledge the big team that's really helped support all this work, as well as funding from NIH. And I think I have a couple minutes here maybe for a few questions. Thanks so much. Yeah.
Depression is such a large category of things. How did you decide what characteristics should go into your program to identify different aspects? Is there some standard out there?
Yeah, so the question is, depression is such a sort of big, you know, I'm adding this heterogeneous thing, and like, how do we think about the categories of things to put into that? I think we really try to think about making sure the different features are combine a lot of the different types of symptoms we might see within depression to make sure that we can look at sort of individual symptom aspects. And I think there's a lot of really nice work developing in this area that's showing like thinking about individual symptoms rather than depression writ large is a much more effective way of going about doing this. I think similar on the intervention side, I think that's actually an important aspect in terms of how we modularize or why we think about these different intervention strategies. And I'll say as a clinical psychologist, very few people, if anyone, has ever come into my office complaining that they're depressed. They come in because they're having trouble sleeping, they're not getting along with people, they can't get out of bed, they don't want to go to work. And so I think that's one thing that we've been doing with a lot of these interventions is trying to develop them to be more focused on the types of things that bring people into the treatment room. Loneliness, amotivation, things of that nature. And I think that having this sort of modular approach allows people to really find something that speaks to why they might seek care.
Then I have a second question. Of all the depressive symptoms, the worst are probably suicide.
Yeah.
And those are something that people don't run up to often, and people around them don't realize that depression has gotten to such a state that these people are thinking about suicide or actually even in suicide. Does this approach help with suicide?
Yeah, so the question is, does this approach help with suicide? There is some work that's been looking at sort of identifying suicidal ideation and non-suicidal self-injurious behavior. I think it's been interesting. I think one of the things that I would still say is that there's not as much work there. And so I think in places like depression, anxiety, both on the intervention as well as now in the digital phenotyping, that's really where that work started. And I'd say that the work in suicide and non-suicidal self-injurious behavior is a lot more nascent, but I think it's promising. I think actually one place where I've been really interested is that box I said about sort of identifying people. There have been sort of people trying to look at like social media data or other things that maybe can reach out to a person when they're starting to show some things, because I think one of the big challenges we see with suicide is most people who who die by suicide have not been seen by a treatment provider recently, and most people who've been seen by a treatment provider recently who die by suicide don't say anything about it to their treatment provider. So trying to figure out some ways to bring in more information into the care pathways, I think, is really important. Thanks. Oh, question here.
I had a question about the insight. I know you spoke about leaving the house being one of the things that you could use the computer to see if they are depressed or not. I was wondering about like what I call with my little brothers doom scrolling, like when they're like just nonstop. And then, is that something that you've looked at?
Yeah, so doom scrolling and of course like social media and mental health is like the hot topic of the moment, especially this week with some different things that are coming out. So we've looked a little bit at this. I mean, I think one thing that we've seen in our research is that it doesn't seem like social media is leading to a sort of big impact on people's mental health symptoms. I think more to what you're speaking to is it does seem like individuals who experience different symptoms use social media differently. We haven't looked at doom scrolling specifically, but I do think like it's interesting to think about sort of two aspects of that. One is, the actual activity that a person's doing with that scrolling behavior. And there have been a lot of research groups who are looking at sort of phone interactions. So again, not the content, but the way you do it. And like kind of this mindless scrolling and aspect, could be something that would be interesting to think about along with some of the symptoms of depression. But then also it's sort of this more passive engagement instead of active engagement. And definitely something that we've seen in our work is using your phones more actively in terms of creating, connecting, and curating content seems to be predictive of better mental health. And so I think if you see people using technology, social media in a way that they're not as actively engaged or really doing that sort of creating, curating, and connecting, I think that does seem to be something we see that is related with worse mental health symptoms. So I have a lot of questions now.
So I know that people or drug companies are now using sort of some of these markers that you were describing as essentially like a secondary measure. I mean, do you think we'll actually get to a point where maybe some of these are going to be like the primary outcome measure for treatment trials as opposed to like self-reported?
Sure. So the question is, will we get to a point where some of these are primary outcome measures? So I will say my bias, I mostly think of myself as a depression researcher when it comes to thinking about specific treatment areas and disorder areas. And the fact of the matter is that the fact that our gold standard measure of depression is a 9-item self-report questionnaire that was created by a drug company is a little head-scratching to me when we have potentially new and better instruments available to us. So do I think that a sort of passively sensed marker of sleep could be a better outcome measure than a self-report questionnaire asking me how I slept last week? Yeah, I think we could get there. I think in the near term, I'm a little skeptical, and especially thinking about, this sort of aspect of going back to this question about, symptom profiles versus overall disorders. I think if they're really targeted to specific things we can learn from this passive data, that's where I think like the real benefit is, but I do think we need we're not there yet. So maybe in the future we'll get there, but I think in the near term, we still have a lot of work to do with developing these digital phenotyping.
You talk about two aspects, detecting and treating.
Mm-hmm.
Let's start with the second one here for a minute. People who need those apps may very well be anhedonic or hypotonic or whatever.
Yeah.
I mean, essentially, how do you get them to stop? And the related question is, do you envision a future in which you track everybody's digital phone and people who have a specific cluster of quote-unquote needing mental health, you're going to actively send those apps to them so that they will get cured without anything else?
Yeah, so it's two questions around sort of the intervention side. One is like, how do we deal with the fact and depression of people who are anhedonic and amotivated and not likely to engage? And then the second question was, if we're able to do sort of wider spread monitoring, might we live in a world where people might push certain interventions at folks? I think the engagement problem is a big one. And I do think that's one of the reasons why a lot of these interventions need some level of human support. Sort of our early work really conceptualized human support mostly to be focused on engagement. This was the supportive accountability model, and that having even the perception that someone was there seemed to be an important aspect of getting people engaged with these programs. So I don't think we're going to move to a world where we're able to have completely unguided programs that are really helping people. This is one reason why in this R01 that I talked a little bit about, that we're using these community human supporters, because the professional support is really not scalable. But if we can use sort of well-meaning and trained and individuals, we might be able to think about creating a more scalable workforce. So yeah, I agree with you. I think we really do need the human component to help with the motivation factor. With trying to push things towards people, it's a real interesting question. And I think one of the places, like as an example, like Google does PHQ-9 screening now, and they don't have as much sort of interventions coming from it, but I think that's a place we can go. We're actually working with Mental Health America, who has a large online screening platform. They screen about 3,000 to 5,000 people per day. And we're trying to develop very brief interventions that once people screen positive on their platform, that we push. We're calling them, it's screening to support or DIY do-it-yourself tools, but we're pushing sort of brief interactions at them that might be able to hopefully start getting them engaged in some different type of care. And then maybe if they need more treatment or more intensive resources, pushing them to resources that might be beneficial for them. So I do envision a world where we're better able to think about the different places where we might come in contact with people and push sort of effective interventions at them.
I think there's so much immense potential in what you're sharing, and I'm wondering if you could speak a little to how we might balance the potential for good with the potential privacy and informed consent concerns that might come along with using the data that's kind of automatically collected.
Yeah, so I think the automatic data collection is really interesting, and as you mentioned, brings up a lot of privacy security concerns. One of the places actually our group and working with some other colleagues here at UCI, like Candace Odgers, has been also really spearheading a lot of this work too, is trying to set up like data donation paradigms. So in fact, like Google collects all this information about you right now, and they have it. And so they also have different capabilities to be able to provide that data to you, mostly for GDPR compliance. Google's is a system called Takeout. Meta has a similar system. And so one of the things that we're trying to work on is better working with some of these different technology teams that have developed these toolkits mostly for GDPR compliance, to think about how we might be able to use those as researchers in data donation paradigms, so that individuals might be able to choose which data they want to share with you. So, can you have my YouTube data? Can you have my search data? Can you have my geolocation data? It's possible in Purple Robot. We actually do have a way where you could turn off individual features if you wanted to. It's just a little bit more clunky. So I do think these data donation paradigms that provide more autonomy to the individuals who want to participate around what type of data they want to share is a really interesting way to think about moving forward with this. But I think a lot of the way that we've been able to, we do a lot of education with our participants. In fact, in the work I mentioned where we were working with youth experiencing homelessness, we did a lot of focus groups leading up to before we did the sensor-based data collection with them to sort of understand what are the challenges, what were they comfortable with, what were they not comfortable with, how can we communicate this to them. So there's a lot of work that really goes into the privacy of ethics, but one of the way we've been able to get this approved by IRBs is really demonstrating that the tech companies have this about you, and they're usually using it for purposes that are not as benevolent as the ones we're doing in research. So, but it's a really good question, something that really does have to be taken very seriously as we do this work. So thanks so much.
Thank you. Okay, now it's lunch. And we'll see at lunch in that